Okay, well, hello to everyone. It's certainly strange circumstances to be delivering this talk, and I hope that everyone is staying well and safe. I would like to begin by thanking OHBM for the chance to give this talk and trying to keep us all connected in these uncertain times. So today I'll be talking about some of the work that we've been doing trying to understand brain network hubs. Now, hubs are highly connected regions of the brain. There's been a lot of work out there showing how important they are for integrated brain function. And in our own lab, we've been interested in understanding how they emerge and what might be so special about them. So today I'll present some of the work that we've been doing, trying to tackle these questions from the perspective of mapping studies, modeling studies, and more recent work we've been doing trying to unpack the genetics of hub connectivity. Now, I just want to say I hope that I'll have a chance to interact with anyone interested in this work throughout the meeting, but if I don't, please do feel free to reach out to me via email or Twitter. The details are there, and I'd be happy to discuss these findings with you further. So the starting point of this work is really the view that the brain is a network. It's probably the most complex network that we know of. It comprises around 86 billion neurons connected by trillions of synapses. And so if we accept that the brain is a network, then an important first step in understanding how the brain works involves mapping how different parts of the brain connect to each other and communicate. And so this led Olaf Sporns and colleagues in this seminal 2005 paper to call for a map of the human connectome, which is essentially a structural wiring diagram. Now, the brain's inherently a multi-scale system, and we can generate wiring diagrams or connectomes at different scales. So at the micro scale, we can map networks of individual neurons and synapses. At the meso scale, we can map how populations of neurons are connected by axonal fibers. And at the macro scales that we typically deal with in human imaging, we can map how large-scale brain regions are connected by white matter bundles. Now, at each of these scales, whenever we obtain this complex map of connections, we're faced with a fundamental question. That is, are these connections formed at random, or are there some underlying principles that govern the way in which brain regions connect to each other? Now, over 100 years ago, Santiago Ramon y Cajal was perhaps the first to propose what are some of the most general and long-standing principles of neuronal organization. He'd spent countless hours peering through the microscope, studying structures of neurons, and from all those observations, he concluded that all of the various conformations of the neuron and its various components are simply morphological adaptations governed by laws of conservation of time, space, and material. Now, personally, I find that to be quite a bold statement. He's saying that all of the brain's complexity can be summarized simply as a need to conserve these three simple properties. But, Cajal is Cajal. He knew his ways around the brain. So, it's probably worth digging a little deeper and trying to think about what these laws might mean for brain organization. And more specifically, we can frame these laws in the language of modern network science, and we can ask, well, what would a connectome look like if it was trying to satisfy these conservation laws? So if we were to try to optimally conserve space and material, we would end up with a network that looks like this. So each part of the brain would connect only to its nearest spatial neighbors. So all of the connections would be short range, and the overall volume of wiring in the network would be quite low. And so we're conserving the cellular material and the space required to link up the network. And this is a good thing because the brain's a metabolically expensive organ, and we want to keep that metabolic expenditure down wherever possible. But we don't do a good job of conserving time. You can imagine if I want to send a message from the back of the brain to the front, I need to go through all of these intermediary connections to do so. It's not a very efficient or rapid way of propagating signals through the network. So we can say, okay, well, what would a connectome look like if we were trying to conserve time? And it can be shown that just having random connectivity does a pretty good job of this. You keep the average number of connections between any pair of regions quite low. But this does come at a cost. We start to see the emergence of these long-range projections that cut across the extent of the network, and this increases the overall volume of wiring in the network and pushes our costs up. So we have a bit of a trade-off here. On the one hand, we can conserve space and material, but that comes at the expense of time. On the other hand, we can conserve time, but that does incur a certain wiring cost. Now, it turns out that brains occupy a space somewhere in between these two extremes. They have mostly short-range connectivity, which helps to conserve material, but they do have some strategically placed long-range projections that act to reduce communication times throughout the network. So it's really not about optimizing any individual conservation law, but really striking a balance between these trade-offs between the three conservation laws. And there's been a fair amount of evidence coming from the work of people like Danny Bassett, Ed Bullmore, and Olive Spawns to suggest that the brain does a pretty good job of negotiating this trade-off, meaning that its wiring cost is near minimal relative to the level of complexity that it requires. So for the rest of the talk, I want to make a case that it's really the connections between the network hubs that play a critical role in determining exactly how nervous systems negotiate trade-offs between Cahal's conservation laws. And I'll use different types of evidence to make this case. And so I'll begin with some work from mapping studies. And by mapping studies, I really mean work that builds connectomes and then tries to describe their properties. So there's a pretty well-established pipeline for mapping connectomes. The first step involves parcellating the brain into different regions to act as our network nodes. And there's lots of ways of doing this. And the selection of a specific method really depends on the questions you're interested in. But once you do that, we then map some aspect of structural or functional connectivity between every pair of regions. And I just want to point out that these processing pipelines are quite complicated and involve a lot of choices and steps, and those choices do affect your final results. We've done a little bit of work on this topic. This is an example of a study done a couple of years ago by Lyndon Parks, who was a PhD student at the time. He's now working with Danny Bassett and Ted Satterthwaite, where he compared a number of different denoising pipelines for resting state fMRI and found that they varied quite a lot in terms of how well they were able to mitigate motion-related confounds in connectivity estimates. And this has a very practical impact, so it can affect where you think connectivity differences are between, say, patients and controls. Earlier this year, in work led by Kevin Aquino and Ben Fulcher, we looked into the properties of global signal regression and found that that might not behave exactly as you think, and proposed some alternatives. And in more recent work led by Stuart Oldham, who's a PhD student in our lab, and this was posted on bioRxiv about a month or so ago. Again, we compared lots of different processing pipelines for diffusion imaging and found, again, that they vary quite a lot in how well they can deal with motion-related contamination. But the good news here is that if you use FSL's latest version of EDDI that includes within volume motion correction and outlier replacement, you can do a pretty good job of mitigating these motion confounds. Now, each of these studies involved a lot of work, a lot of data, and so I don't have time to go into the details, but anyone interested should definitely check it out and think about their pre-processing pipelines carefully. And I'll also say that I'm being extraordinarily self-centered by only talking about work in our own lab. A lot of people have done some really excellent work in this space. I've just included some citations at the bottom of the slide there, particularly to work from Ted Satterthwaite and Jonathan Power's teams that's been influential in our own thinking. But assuming that you're happy with your pre-processing pipeline and that you've mapped connectivity between every pair of brain regions, you can then represent that connectivity in the form of a matrix where each row and each column encodes the type and strength of connectivity between regions. And then from this, we can build a graph-based representation where each region is represented as a node and we draw an edge between nodes if they're connected in some way. Now, once we have this graph, we can do two broad classes of analysis. So one is focused on looking at variations in the strength and type of connectivity at each edge. And the other is focused on understanding network topology. And it's really these topological analyses that try to understand how connections are arranged with respect to each other in the network. And this is where we use the mathematics of graph theory to derive a number of different quantities that try to capture how the network is organized. Now, there's been a lot of work that's gone into trying to understand what are the key topological principles of brain networks, and I'm going to boldly try to summarize that with a single slide. But so some of the main properties are, the first is that the brain can be divided into subsets of nodes called modules, and so these are strongly interconnected with each other. They're represented by different colors here, and so it's thought that they serve some kind of common function. So you could think of them as a visual system, an auditory system, and so on. Now, most of the nodes within a module are located near each other in space, so a lot of the connectivity is short range. So this organization promotes segregation and specialization of function, but also helps to conserve material, going back to Cahal's laws. Now, each of these modules has one or more hubs, and these hubs are strongly interconnected with each other, forming what we call a rich club. It's like its own module that overlaps with the more specialized modules. And the idea is that hubs are rich in terms of the number of connections they have, and they're a club because they're strongly connected with each other. Now, this rich club organization is thought to conserve time and promote rapid integration across the network. You can imagine if I want to send a message from this node to this node here, I need to go through this rich club of hub connectivity to do so. Now, hubs and rich clubs have got a lot of interesting properties that suggest they really play a key role in how the brain negotiates trade-offs between Cahal's conservation laws. And so I'm going to dwell a little bit on how we define them. Now, assume that there's lots of different ways to define hubs, but assume we take the simplest possible measure, which is just node degree, and that's the number of connections that's attached to a node. We can determine whether a network has a rich club or not using this phi coefficient here, what we call the rich club coefficient. And this is just the density of connections that we see in the network between the hubs. Now, the critical variable in this equation here is K, and K is our threshold for defining hubs. So should we take a node with more than 10 connections as a hub, or 20 or 50? It's unclear. And so we compute this value across a range of plausible thresholds. So just to make this a little bit more concrete, here is a schematic of the mouse connectome. And let's say we start with a liberal threshold. So, okay, I'm going to take all nodes with more than 2 connections and call them a hub, marked in orange, and all other nodes are non-hubs, marked in blue. Now, an interesting observation that was first made by Martin van den Heuvel and Olaf Sporns a few years ago was that once you distinguish between hubs and non-hubs, you have 3 types of connections that occur in the network. You have what are called rich links, which are connections between hubs. They're shown in red. You have feeder links, which are connections between hubs and non-hubs, shown in green. And you have peripheral links, which are connections between 2 non-hubs, shown in blue. I'm going to refer to this distinction quite a lot throughout the talk, so it's important to keep in mind. At this stage, I just want to point out that the way in which we make this distinction very much depends on our hub threshold. So let's say I take a more stringent threshold. We say, okay, all nodes with more than 3 connections are hubs. Then we end up with just 4 hubs in the network, fewer rich links, and proportionally more feeder and peripheral. And again, if we become more stringent and say, okay, all nodes with more than 4 connections are hubs, then we just have 2 nodes and 1 rich link. So when we take this to actual data, in this case here, we see the degree distribution of the mouse brain. So remember, degree is the number of connections attached to each node. And here we see how that's distributed across all nodes of the mouse brain, different regions. So you can see there's a long tail here, which is consistent with the idea that there's a subset of nodes that are quite strongly connected. But it's also unclear where you should distinguish hubs from non-hubs. Should I just take this single node out here as a hub, or maybe this group, or this group, or this group? It's unclear. So we start with a very liberal threshold down here at the bottom of the distribution. So everything above is a hub, nearly everything in the network is a hub. And we compute our phi coefficient, and then we do this for all possible hub thresholds. So moving through the distribution. So we plot the rich club coefficient here, and here it's normalized by what we see in an appropriate null model. So values greater than 1 are indicative of the fact that we see greater connectivity between the hubs of the network than we expect by chance. So you can see we see a sharp increase in this coefficient at a degree threshold of about 42. You can see we're right in the tail of the distribution here, so this really is corresponding to the hubs of the network. And this gray area here is what we call the topological rich club regime. So it's where we see statistically significant evidence of rich club organization of the network. And then the values drop off at the bottom because once we get right out into the tail, we don't have a lot of hubs and the statistics get a little noisy. So we've got evidence of the rich club organization in the mouse. We can then ask, well, where are the hubs in the brain? And so here, different anatomical divisions are represented with different colors, and the area is proportional to the number of hubs that are in each division. You can see we find them right throughout the mouse brain. We've got them in cortex, hypothalamus, midbrain, medulla, and so on. So they're not restricted to any single part of the brain. Then you can say, okay, well, if that's the case, if hubs are kind of scattered throughout and they play an important role in integrating these disparate networks, then we would expect them to have quite long-range connectivity. So their wiring costs should be quite high. So here we've got three different measures of wiring cost. This is analyses done by Ben Fulcher a few years ago. We've got the average connection distance between rich links. We've got the probability of finding a reciprocal connection between hubs, and the average connectivity weight between hubs. And you can see in all cases, they increase as a function of our degree threshold, indicating that the wiring cost is highest for the connections between the most highly connected nodes of the network. And so I'm going to show this kind of plot quite a lot. And so the basic idea is whatever property we have indicated on the y-axis here, if it increases as a function of x, it means that it's more strongly expressed in the hubs of the network. So these data are just indicating that those connections between hubs are among the costliest in the mouse connectome. We can then say, okay, well, they're quite costly. They must be doing something to justify this cost. So we can estimate measures of edge centrality. These try to quantify how important a connection is for communication in the network. So here we've got two different measures of centrality. They just make different assumptions about how communication happens in the network. And in both cases, they increase as a function of our hub threshold. So again, consistent with the idea that those connections between hubs are the most central. Now, this is some data from our own analyses in the mouse brain, but this is not new, this idea that connections between hubs are very costly but functionally valuable. This has been shown quite a lot in a lot of different species with connectomes mapped using different techniques. And I've got some citations here. A lot of this work has been led by Martijn van den Heuvel and Olaf Spons. Now, in humans, we can get converging evidence of the high cost of hubs from metabolic imaging. So this is some data from Marcus Reichel's team where they mapped areas showing high levels of aerobic glycolysis. And it turns out these areas correspond quite nicely with what we think to be the default mode network and kind of executive control systems. These are largely areas of association cortex, which we know to be the hubs of the brain. And it turns out that this and so this is consistent with this idea that hubs are really highly active, involved in integrating information from different parts of the network. And it turns out that this high metabolic demand might make these areas especially vulnerable to the effects of disease. So in a study done a little while ago from Randy Buckner's team, they showed areas of high amyloid deposition in people with Alzheimer's disease. And they found that those areas correspond quite nicely with the hubs of the brain. And in a more recent meta-analysis from Nicholas Crossley and Ed Bullmore of VBM studies across 26 different psychiatric and neurological conditions, they found that for the majority of those, grey matter volume reductions were more likely to occur in the hubs of the connectome. And these are very different disorders. So you've got autism, schizophrenia, Huntington's disease, depression, epilepsy, and so on. So it suggests that regardless of the underlying causal mechanisms, pathology tends to accumulate in the hubs of the brain. And there's different reasons for this, the high metabolic demand being one of those. So if we accept that rich links between hubs are among the costliest and most functionally valuable connections in the brain, then it seems reasonable to say that they really play an important role in determining how a given nervous system negotiates trade-offs between Cahal's conservation laws. So then what drives the emergence of hubs and rich clumps? We can get a bit of insight into this using generative models of brain network wiring. And the idea in this type of work is to grow networks computationally according to specific wiring rules and see if we can obtain networks that look like an actual connectome. Now, the most successful class of models for the human connectome that have been considered thus far have this general form here. And this tries to capture this trade-off between cost and value. And so I should point out that this all comes from some really seminal work by Petra Vertesh and Rick Betzel. So the model indicates that the probability of connecting any two nodes, U and V, is proportional to the distance between them and some topological property that is a proxy for functional value. Now we see that there's a negative exponent on D here, meaning that we're less likely to form long-range connections, so consistent with the idea that longer connections are more costly. And there's a positive exponent on T, indicating that we're more likely to form connections that are topologically favorable, that provide us some kind of functional advantage. And the values of eta and gamma really determine how strong these biases are, and these are fitted to the data. So you might say, okay, well, what exactly does functionally valuable mean, and what is T? And that really depends on the hypothesis. So lots of different properties have been substituted for T in this equation. Here's an example of just some of them. But in head-to-head comparisons, Rick and Petra have shown that one single property, the matching index here, seems to do quite well. So here, these are estimates of model fit to actual empirical connectomes of different models, and you can see the matching index is doing best here. So lower is a better fit here. And so under this model, connections are formed at random, subject to the constraints that we're less likely to form long-range connections, but we're more likely to form connections between two nodes if they connect to other similar nodes. So in a very general way, this mechanism mimics a kind of Hebbian-like plasticity process. Now here is an example of how the model actually fits the data. So the solid lines are the empirical distributions across nodes that we see in the connectomes. This is the degree distribution expressed as a cumulative distribution function, distribution of clustering coefficient, betweenness coefficient, and distribution of connection lengths across edges. And the light lines are different runs of the model. And you can see they do a pretty good job of capturing these distributions. And so it seems that these synthetic networks that we grow according to this model have got a lot of the sort of complex topological properties that we would normally think of as important for the brain. So we might say, okay, well, job done. We can grow something that looks a lot like the brain using this very simple model with only 2 free parameters. But things are a little bit more complicated. And this is work currently underway in our own lab, led by Stuart Oldham and Arina Adnatkevichute. And we found that the spatial embedding of connectivity in the model and data is very different. So here is a map of actual hubs in a human connectome generated with diffusion MRI. And here is a map of hubs in the model. So you can see in the model, they all cluster near the center of the brain around insular and auditory cortex, whereas there's more of a widespread distribution in the actual data. So if you actually plot the correlation of the degrees between model and data, you get almost 0. And in fact, across 10,000 different runs of different models with different parameter values, we rarely get a correlation that exceeds 0.2 or 0.3, even if we're directly trying to optimize this correlation in our parameter fitting. So what these results suggest is that while these stochastic models do quite a good job of capturing the general statistical properties of the brain, they do a poor job of capturing the way in which these properties are spatially embedded. So it's not actually capturing the specific way in which hubs and rich clubs are wired. And this is important. And for those who are interested, I've got some citations there to some lovely work by Leo Goyer, James Robertson, and Michael Breakspear, where they've shown using a very different analytic approach that where hubs are in the brain matters for dynamics and the complexity of the dynamics that can emerge. So if we can't explain the emergence of hubs and rich clubs, at least as they're realized in the brain through simple stochastic processes, what might explain their emergence? Well, genes would be a logical first guess. We know that they play an important role in shaping brain connectivity early in development. And in our own work some time ago, we found in a small scale twin study some preliminary evidence where we derived a measure that tried to capture the cost efficiency of each brain region's connectivity, and found that that was quite strongly heritable, particularly in the hubs of the brain, especially areas of default mode and frontoparietal systems. And so for the rest of the talk, I'm going to talk about how we've gone about trying to unpack the relationship between genetic function and hub connectivity over the last few years. And so in this work, we're really trying to link molecular function to connectome properties mapped at each of these scales. So we've done work at each of these scales. And I'll begin by talking about some work we've done in the mouse at the mesoscale. And this was work that was done by Ben Fulcher a few years ago when he was a postdoc in our team, and he's now based at the University of Sydney. So in this analysis, we used data from the Allen Brain Institute. So a few years ago, they published a draft of the mouse connectome generated from a large number of track tracing studies. And they have also made available an atlas of gene expression in the mouse brain. So for each of over 200 brain regions, we have expression measures in over 17,500 genes. That's shown on the right, the connectomes on the left. So in our analysis, we distinguish between hubs and non-hubs, and then we map the rich feeder and peripheral connections. So remember, a rich link is a connection between two hubs. A feeder is a connection between a hub and a non-hub, and a peripheral is a connection between two non-hubs. So then for every pair of regions, we've got the type of connection they're linked by, and we can then go to our gene expression matrix, pull out the expression profiles of each node, and then compute the correlation between each pair of nodes. So we have a measure of how tightly coupled their gene expression profiles are. And then we can just ask, well, does this level of transcriptional coupling vary as a function of our link class across rich, feeder, and peripheral? So this is what I've shown here. On the y-axis, we've got the average correlated gene expression or transcriptional coupling between a pair of nodes. And we see how this varies across our different thresholds for defining hubs. You can see across almost all thresholds, the transcriptional coupling is highest for rich, intermediate for feeder, and lowest for peripheral, with a bit of a bump here when we get to the topological rich club regime. Now this image just shows the anatomical distribution of the findings. The details are not so critical, but the basic idea is it's not an effect that's confined to any single anatomical division, but is related to the hubs right throughout the brain. So that was interesting, but it's a fairly coarse summary. We're taking data from 17,500 genes and summarizing it with a single correlation estimate. So we developed a way to score the contribution of each individual gene to that correlation estimate. And then we ran tests for enrichment to determine whether certain functionally related groups of genes, as determined by the gene ontology annotation system, are on average show a higher contribution to this effect. And when we do that, we find evidence for significant enrichment of 5 gene ontology categories that are related to the synthesis and breakdown of ATP. So in this very data-driven way, we've been able to pull out this molecular signature of the high metabolic demand of hub regions. Now, I just want to point out that if you look more generally across the brain, forget about hub, non-hub, you look at all connections, you find that the probability of any two nodes being connected declines quite sharply as a function of their distance, as does the strength of their correlated gene expression. Whereas what we find with hubs is that on average, they're separated by longer anatomical distances, they're strongly interconnected, and they have among the highest levels of transcriptional coupling. So really suggesting that there is something quite distinct at the gene expression level about pairs of hubs. So that was nice, but we wanted to see if we could see this in another animal. And so we turned to data in the nematode worm C. elegans, and this is a famous animal in connectomics. It was the first to have its connectome mapped entirely at the level of each and every neuron and synapse using electron microscopy. So the data for an imager are as real as they get. And we also know a lot about the individual properties of neurons. Each neuron has its own name and a web page, and we know things like the neurotransmitters they secrete, their birth times, and so on. And so we can ask questions about what properties might be driving the expression findings. This is work that was led by Ben Fulcher together with Arina Adnatkevichute, who was a PhD student in our lab at the time. So on the left here, we've got the connectome for the worm, and on the right, we have a gene expression matrix. It's not as nice as for the mouse. We only have data on 948 genes, and it's based on the curation of published findings in the literature, and it's binary. So we only know if a gene was expressed in a neuron or not, but we don't know by how much. But it's enough to get started. And the nice thing is we do have all this neuronal metadata. So we've got neurochemistry, anatomical position, birth time, and so on. So here is a map of where the neurons are in the worm, and all the hubs are located in the head and the tail. So we get these long-range projections that extend across the body of the worm, consistent with the idea that those rich links are the costliest in the worm's connectome. And this was something that had already been shown by Emma Tolson and Ed Bormore. And if we repeat our gene expression analysis, we get the same basic pattern. So coupled gene expression is highest for rich, intermediate for feeder, and lowest for peripheral. To dig a bit deeper, we can cut these data at a specific hub threshold and just plot the distribution of correlated gene expression estimates across all edges in each category. So here we've got the distribution for rich links, different types of feeder links, and peripheral. But then we can look at other pairs of regions in the worm. And so here we can look at correlated gene expression for unconnected pairs of neurons, unidirectional chemical synapses, reciprocal chemical synapses, and gap junctions, so electrical synapses. And in all cases, you can see the average correlated gene expression is highest for rich links. So again, consistent with this idea that there is something quite special, even at the neuronal level, about connected pairs of hubs, as reflected by gene expression patterns. Now, Ben and Arina then went and did a lot of careful analyses looking at this neuronal metadata to try to understand what might be related to this effect. And they were able to show that it's not related to the fact that all hubs might be motor neurons or sensory neurons or anything like that. It's not related to similarity in their anatomical position or the neurotransmitters they secrete. It's not related to the topological module they belong to or similarity in the birth times at which when these neurons are born. Nor is it related to the lineage distance. So this is the number of cell divisions that separates a pair of neurons from a common progenitor. What it does seem to be related to is the fact that most hub neurons are a specific class of interneuron called command interneurons. So when you separate the hubs into command interneurons and non-command interneurons, you see quite a big difference in the correlation estimates. And command interneurons play a really important role in regulating locomotion in the worm, which, if you think about it, is one of the more complex behaviors the worm can engage in. So in a sense, there's a bit of a parallel here with what we see in humans, where areas of association cortex are hubs. And so it suggests that network hubs are really involved in the most complex behaviors of a given organism. So what these results collectively suggest then is that this transcriptional similarity between hubs is not related to anatomy or chemistry or developmental proximity, but may instead be related to the actual functional roles of the hubs in a network. So it's quite nice to see this replication of findings across worm and mouse, but then of course it does raise questions about what we see in humans. And so that's what I'm going to focus on for the rest of the talk. And again, this is work that has been led by Arina Adnatkevičiūtė, which she did as part of her PhD. And we hope to have this available on bioRxiv within the next few weeks, if it's not already on there by the time this talk is broadcast. In the first instance, we turned to data again from the Allen Institute. So they have also made available an atlas of gene expression in the adult brain. This is constructed from microarray analysis of nearly 4,000 tissue samples. So on the left here, we see the locations of those samples taken from 6 postmortem brains. So in each sample, we've got measures of expression in over 20,000 genes. And this is a complicated figure. It just shows different ways that you can relate the expression data to imaging measures. But for our analysis, we just did the same thing we did with the mouse and the worm. We map the samples to brain regions. We construct a region by expression matrix. And for every pair of regions, we compute the correlation and expression profiles and look at that across our different link categories. I just point out that these expression data are not necessarily plug and play. They do need to be processed with multiple steps that require choices to be made, and these choices can affect your final results. And so for anyone interested, Arina went into a lot of detail trying to understand the impact of all of these choices and provided some recommended workflows. And I'll just give a shout out to Ross Markello and Bratislav Misic, who developed a Python toolbox that implements some of these steps. So anyone interested in dealing with these data, I recommend you check this out. So go back to the human. We focus on the left cortex, the left hemisphere, because that's where anatomical coverage in the atlas is most comprehensive. And if we repeat our gene expression analysis, we find the same basic pattern. So coupled gene expression is highest for rich, intermediate for feeder, and lowest for peripheral. And we're able to replicate this in an independent cohort, so a different group of people using a different connectome. And if we do our enrichment analysis, once again, we find that the most strongly enriched categories are those involved in oxidative metabolism. So aligning almost perfectly with what we see in the mouse. Now, there is a caveat, and this is work that we've very recently posted on bioRxiv, led by Ben Fulcher. And it turns out that these enrichment analyses, when you apply them to gene expression atlas data, so spatially embedded atlas data, have certain biases. And these biases, the practical effect of these biases is that you can obtain evidence of significant enrichment even with completely random phenotypes. And more worryingly is if you look across the literature, the categories that most frequently come up as showing enrichment for different phenotypes tend to be the ones that suffer the most from this false positive bias. Now, I don't have time to go into the details, but in the paper we explain why this occurs, and we propose some solutions based on more constrained null models. And so anyone doing this kind of work should check this article out. I just want to point out at this stage, so this doesn't affect the overall finding that coupled gene expression is highest in hubs. It affects the interpretation of which genes might be driving this effect. Now, I'll present some data later to suggest that there is something to the metabolic signal that I've been describing. But we also did want to go a little bit deeper in our own analyses and do something a little bit more hypothesis-driven. And specifically, we were interested in the role of cytoarchitecture. This is because in the Allen Institute, the data come from bulk tissue samples, which mix signals from different cell classes. And so it's possible that you might see differences in gene expression being driven by just differences in the relative proportions of different cell types. So to do this, we teamed up with Gareth Ball at the Murdoch Institute in Melbourne, who had adapted an approach first developed by Jakob Seidlis at NIH, where we identify sets of genes that show cell-specific gene expression. And so we can use these as markers of the relative abundance of different cell types. So here we've got 7 different types. We've got excitatory neurons, inhibitory neurons, oligodendrocytes, microglia, and so on. And so we separate these lists and then we do the same analysis in each list separately. And in all cases, you can see that coupled expression is highest for rich links. So it is suggesting that this overall effect is at least partly related to similarity in cytoarchitecture between hubs. But this is a fairly indirect measure, and we wanted to verify this using an independent approach. And so we teamed up with Casey Pequola at the MNI, and we looked at data from the Big Brain Atlas. So this is a high-res, post-mortem brain, silver-stained, mapped to stereotaxic space. And Casey's developed a way to generate depth-dependent intensity profiles. So we basically fill the cortical ribbon with a series of intermediate surfaces, and we estimate the intensity at each of those surfaces as a proxy for variations in cell size and density. And then we can correlate those depth-dependent profiles between regions. And if we do this, we again find that the correlations are highest for the rich links. So consistent with the idea that connected hubs have got more similar cytoarchitecture compared to other pairs of brain regions. So that's quite nice. So using these gene expression data, we've been able to show consistently across the worm, mouse, and human that hubs have got tightly coupled patterns of gene expression related to metabolism and cellular properties. But these data are required in, the expression data are required in adults, and it's possible that if you look earlier in development, you might find different results. And so it's kind of an open question. Maybe what we're seeing is just a property of the function of these regions, or their actual anatomy in the developed brain. And so it's kind of an open question as to whether how genes actually sculpt hub connectivity. So to get a bit more traction on this, we can do some additional analyses in the human. And so we have done a classic heritability analysis using data from the Connectome project. And so in this case, we fit a structural equation model at each connection in the brain. So over 12,000 connections, well, depending on the parcellation, but we replicate this using different parcellations. And these models allow us to estimate the degree to which genes and environment explain individual differences in connectivity strength. And we estimate connectivity strength using mean tracked FA, but we get similar results if we use streamline count. And so we generate these estimates of genetic and environmental influences and look at how they differ across our linked classes. And so this is what the results look like. And again, we see genetic influences are highest for rich, intermediate for feeder, and lowest for peripheral. And these effects are quite high. So once we're in the tail of the distribution, on average, the heritability estimates are over 50%. So for connected hubs, on average, over 50% of the variance in connectivity strength is explained by genes. And on the flip side, if we look at environmental influences, they are lowest for rich links and highest for peripheral. So these results are suggesting that while individual differences in hub connectivity are mainly driven by genetics, the differences in the connectivity of non-hub areas are largely influenced by environment. So you might say, okay, well, is this related to just network affiliation? Maybe most of the hubs are in one specific network or another. So we assign our regions to different canonical networks using the parcellation from Lee et al. Of the HCP Atlas. And we find that hubs can be found in just about every network, but when you really get to the most highly connected nodes, they are overrepresented in the association networks. So default mode, frontoparietal, singular, opercular. And if we look at heritability estimates for connections within and between networks, we find in both cases they're highest for rich links, so it's not explained by that. And if we look across all 12 canonical networks, we find in no case is the average heritability higher than what we see for rich links. So this really does suggest that genetic influences are most strongly pronounced on those links between hubs, regardless of where they sit in the network, so distributed right throughout the brain. We then wanted to go a step further and try to understand which specific genes might preferentially influence hub connectivity. And this is hard to do without massive samples that you would need for GWAS. But to get a bit of traction, we developed a way for identifying SNPs that influence hub connectivity and then mapping these to the genes that, whose expression they influence. And so I'll just talk about this process in a little bit. We start by running a GWAS at each edge of the connectome using unrelated subjects from the HCP data. And then for each SNP at each edge, we obtain a beta estimate quantifying the degree to which it influences mean tracked FA. Then we sort our connections into rich, feeder, and peripheral and extract the data for the rich and peripheral edges. And then we use that to build an edge by SNP matrix of beta values. So remember, the beta values encode how strongly a SNP predicts connectivity in a given edge. And then we obtain the distributions for rich links and peripheral links for a given SNP, and we compare them using a t-test. And so in this case, higher t values indicate that a given SNP has a higher, is better able to predict connectivity strength for rich links compared to peripheral on average. So we select the top 10% most discriminating SNPs, and we identify which of those are EQTLs, so which of those impact cortical gene expression using data from the PsycEncode and GTEx consortia. We map those EQTLs to the genes that whose expression they influence, and then we repeat this process for SNP lists that we obtain from prior GWASs of intelligence and mental disorders. And we try to test for significant overlap between our hub gene list and the gene lists for those phenotypes. And if we do that, we find evidence of significant overlap between our lists, our hub list and the list of genes implicated in intelligence and schizophrenia, but not depression, bipolar disorder, autism, or ADHD. Now, some of you are probably recoiling in horror saying, how could they possibly do a connectome-wide GWAS in 252 people? And so I'd just like to make 2 points in response. So the first is that we're not trying to conduct inference at the level of individual SNPs. We're just using the GWAS to obtain beta estimates, which we then feed into our EQTL mapping pipeline to obtain the gene lists. And the second is that we repeated the analysis in a completely independent sample, and we got the same basic findings. We find significant overlap with our hub gene list and those for IQ and schizophrenia. So that gives us some confidence that these findings are robust. We then did a more global enrichment analysis, asking if there are any other functionally related groups of genes that are enriched in our hub gene list, and we found evidence for significant enrichment of genes involved in energy metabolism in both cohorts. So some nice converging evidence that aligns with our gene expression findings here. So suggesting that EQTLs for hub connectivities also influence the expression of genes regulating energy metabolism. So to summarize, if we return to Cahal's conservation laws, what have we learned about how they might manifest in the brain? While it seems modular organization helps to conserve space and material, and hubs and rich clubs conserve time, they promote integrated, complex brain function, albeit at some cost. And so the trick is in how the brain negotiates this trade-off between time, space, and material. The topography of hubs, the spatial embedding of hubs is not random in the sense that we can't reproduce it using existing stochastic models. And I've presented evidence to suggest that hub connectivity is under quite strong genetic control. So genetic influences are not distributed homogeneously throughout the brain, but they are most strongly concentrated on those costly and valuable links between hubs. We've also seen that connected hubs show tightly coupled patterns of gene expression that's related to cellular architecture and metabolism, and that EQTLs for hubs also impact the expression of genes related to metabolism, intelligence, and schizophrenia. And so something that we're interested in following up is whether genetic influences on intelligence and risk for schizophrenia are mediated through their effect specifically on hub connectivity. So I'll just close by thanking the people that did the work, especially Irina Adnatkevich-Shurte, who did the human and worm work, and Ben Fulcher, who did the worm and mouse work, and Stuart Oldham, who did the modeling work. There's some links to some of the software we use. Thanks very much to the Allen Institute and HCP for making their data available. And for anyone interested in network analysis, Ed Bullmore, Andrew Zaleski, and I wrote a textbook a couple of years ago, which is available through Amazon and Elsevier. But thank you for your attention.